have discussed definitions of aging and how to measure aging. But we still need to understand why we age in the first place. Sir Peter Medawar famously wrote an essay calling aging an unsolved problem of biology. The problem that biology has with aging is that, from an evolutionary perspective, aging is maladaptive. In evolutionary terms, success is defined as reproductive success, that is, to pass on genes to the next generations. These passed on genes then will shape the next generations. It therefore stands to reason that long-lived organisms have a greater chance of successfully passing on their genes to the next generation, including those genes that make them long-lived in the first place. In other words, longevity should be selected for by evolution. However, clearly, aging and functional decline is a near universal feature. So how can that be? In his essay, Medawar sought to address this question. He focused not so much on the how of the aging process, the mechanisms of the aging process. These are described by quite a few different so-called proximate theories, and we will discuss those throughout the course. Now, he, in contrast, thought to identify the ultimate cause of aging, why aging occurs in the first place, especially in the light of those evolutionary forces that should disfavor aging, as we just discussed. Early hypotheses to explain the ultimate reason why aging persists, despite evolutionary pressures to the contrary, are so-called group theory. The idea that old, non-productive individuals have to be eliminated in order to give rise to more vigorous, younger individuals. However, in evolutionary terms, beneficial adaptations to an individual almost always trump what happens to a group, because that individual reproduces more. The most famous hypothesis to explain aging is the so-called rate of living hypothesis. It states that longevity is directly related to metabolic expenditure, or metabolic rate, the rate of living in the rate of living hypothesis. In other words, the slower the metabolic rate, the longer the lifespan of an organism. At first glance, this makes perfect sense. Mitochondria are the power plants of the cell that produce all the energy that the cell needs, but as a consequence of this activity, they also produce toxic byproducts, so-called radicals, and they are inevitable byproducts of this cellular metabolism. Thus, lower metabolism would mean slower accumulation of these radicals. This part of the hypothesis is present in the radical theory of aging, which we will discuss in a later module. For example, the fruit fly Drosophila melanogaster lives much longer when raised at lower raising temperature. If you raise them at 18 degrees Celsius, they have a mean longevity of around 130 days. But if you increase their culturing temperature to around 30 degrees Celsius, they only live about 30 days. And this is because they are so-called poikilothermic animals that do not control their own body temperature, and this lower temperature simply slows down their metabolic rate, and that increases their longevity. In mammals, there is a clear relationship between the mass of an animal and its longevity. Larger animals, like elephants, usually live longer because their mass-specific metabolic rate is usually lower than that of smaller animals. The bodies of these smaller animals have a larger surface-to-volume ratio, which means they lose body heat much more rapidly and thus have to speed up metabolism to maintain their temperature. So that means they increase their metabolic expenditure, and then this would shorten their longevity. However, the rate of living hypothesis falls apart if you just poke it enough. For example, marsupials generally tend to have lower body temperatures than their mammalian cousins, yet they have shorter lifespans. More dramatically, birds have much higher metabolic rate than similarly sized mammals due to the higher energy expenditure of their flight muscles. Yet they live considerably longer. The common mouse lives for about 2 years, while even small hummingbirds can live up to 14 years. Even within a species, the metabolic rate longevity relationship does not hold up, and the better we get at measuring metabolic rates, the less stringent that relationship becomes. So clearly, the rate of living hypothesis fails to explain why we age. The breakthrough that Medawar made in understanding aging came from his genetic studies of Huntington's disease. HD is caused by a dominant genetic mutation that is invariably fatal. HD is also a late-onset disease, with individuals usually showing symptoms beginning in their late 30s and early 40s, past the time of their peak reproductive age, which is between their 20s and 30s. How can such a disease exist within the human gene pool when it has a lethal phenotype? When, in other words, evolution should easily get rid of that? The answer lies in the age of onset of the disease. The onset is after the peak reproductive period in life, and the disease is asymptomatic before. This means that any mutation that only has effects later in life, after reproduction has ended, is no longer subject to evolutionary forces because the genes have already been passed on, and thus these maladaptive genes that now cause problems this late in life therefore persist throughout the generations. As the force of natural selection thus declines with increasing age, this means that any mutation that acts late in the life of an organism with detrimental effects on survival may be able to persist in populations and will eventually spread throughout populations. This is also known as the mutation accumulation theory, which posits that over many generations, these detrimental mutations accumulate in the genome, which in turn predispose the organism to reduced functionality, and this is what aging is. Another consequence of mutation accumulation is the emergence of diseases of aging, such as Huntington's disease, as we just seen, and other late-life diseases such as cancers. A further development of the mutation accumulation theory is the antagonistic pleiotropy theory. It states that some mutations may have beneficial effects early in life, leading to increased early reproductive success. If such a mutation, however, has pleiotropic or additional effects, especially late in life, then once again, the forces of natural selection would not act on this mutation. They would even favor it because it's beneficial early in life, and that leads to its persistence in the gene pool. If these pleiotropic, late-acting, late-life effects are detrimental, then they may contribute to late-life decline and thus contribute to aging. This situation represents a trade-off between the life history traits longevity and fertility. That is, if something is beneficial for reproduction and health early on in life, it may be detrimental to lifespan late in life. One such example for antagonistic pleiotropy is the p53 gene. p53 is one of the most important tumor suppressor genes of the genome, and it protects the organism against the development of cancer. As such, it is clearly beneficial early in life. It protects the body from development of cancers that will kill you. Disabling p53 leads to massive cancer development and early death. At the same time, p53 does promote aging, and disabling p53 late in life does in fact extend longevity, as shown here from the data in Drosophila. In these animals with normal p53 function, you have a normal longevity, but if you disable p53 late in life, you can actually extend the lifespan of these fruit flies quite dramatically. We will talk more about the cancer longevity trade-off in later chapters, and especially about p53. Many other examples for antagonistic pleiotropic trade-offs exist, and most are in fact affecting reproductive and immune systems. For example, the genes listed in this table with examples from C. elegans, the nematode roundworm, the fruit fly Drosophila melanogaster, or even from mouse models, the mammalian models. You'll see many genes, many genetic interventions that increase longevity, but they are accompanied by a pleiotropic trade-off, especially in the reproductive system and sometimes also in the immune system. The final evolutionary theory that I want to discuss today that attempts to explain why aging exists and why trade-offs often exist, especially within the reproductive system, is the disposable soma theory. The basic premise of the disposable soma theory is that non-germ cells, the somatic cells or soma, are only there to support the germline and to aid in the transmission of the genetic material to the next generation via germ cells. After that is achieved, the soma are no longer required and thus disposable. This theory works on the concept of resource allocation. As there are only limited amount of resources available to an organism, it needs to allocate them between the germline and the soma. An organism can choose to allocate resources to the maintenance and repair of the soma, which keeps those cells healthy and for longer periods, but that would come at the cost of insufficient resources for the germline, and thus lower reproduction. Or vice versa. Resources can be shifted towards reproduction, but at the cost of decreased somatic maintenance, and thus shorter lifespans. These evolutionary theories are not exclusive to each other, and often a combination of them is needed to explain aging in a particular species. Sometimes these theories fall short of explaining aging patterns at all, for example in single-celled organisms, like these yeasts shown here, or organisms that do not have a clear distinction between germline and soma. They can, however, serve as a starting point for experimental dissection of the aging process and hypothesis testing. They thus enable a researcher to finally ask the question of how aging proceeds. Check your understanding. Explain aging in evolutionary terms. Compare and contrast the different evolutionary theories of aging.